but certainly not least today, we have Bailey Engle. So Dr. Engle is a research geneticist at the USMARC, focusing on genetic selection and improvement in beef cattle. She's trained at Montana State University with a BS in animal science and Texas A&M for her PhD in animal breeding. And her research interests center on utilizing a combination of molecular and quantitative genetics to better understand genomic trait architecture and to increase the effectiveness of genetic selection. Her research program is largely focused on genomic selection for cow and heifer productivity, but she also has some experience working with some groups that also do metagenomics work. And so that's the expertise that she's bringing here today, and a bit of a twist on what she works on a lot of the time. So really excited to have Bailey here. I'm gonna hand it over to her.
Thank you, Megan. Like she said, I'm a quantitative geneticist, which means I am not a microbiologist, and I'm also not an expert in metagenomics. But this is something that I'm really interested in, very excited about. As she referenced, I've worked within groups that are actively engaging in these projects, and I'm working on developing projects of my own in this space. As some of you guys may know, I've only been at the USMARC for a little over a year, so working on this. But regardless of that, this is a new and emerging topic within our field. And so with that, it means that there's not really many experts on this topic globally. So does that make me qualified to speak on it? I don't know, but I am very honored to be able to share with you guys something that I think is quite interesting, something that I think holds some potential, but is also under development, which for those of us who do research, I think poses an opportunity. So metagenomics. First, we'll talk about what even is metagenomics and why you guys might care about it. And then I'll go into what I think the opportunity is. I want to preface that this is my perspective of where the opportunity of metagenomics might fall. And I'm confident that if you asked a microbiologist, or maybe even somebody who focused very deeply into the ecology of a metagenome, they might have a different answer. But again, I'm a quantitative geneticist, and so we're going to be talking about metagenomic prediction. And I will share with you guys at least a little bit of an overview of what that looks like in the event that you might want to investigate it yourself. I'll share my perspectives of where I think there's future opportunities, and then I'll also share where some of the challenges and ongoing questions lie. Again, for those of us in the room who do research, I hope that this is of interest to you and might inspire you to consider it further. So just to make sure we're all on the same playing field or on the same page, we're going to start with some basic vocab words. So first, what is the microbiome? And that is just the community of microbes. We might think about it as the skin microbiome. What microbial communities do we find on our skin? Very often we think about the rumen microbiome and what is the composition of the microbes that make up the rumen. So when we want to investigate that, we'll often use investigate the metagenome, which is really just if we were to sequence every single microbial cell within that microbiome, we would be getting the metagenome. So it's just sequencing microbes. And then the investigation of the metagenome is metagenomics. And so why do we care about metagenomics? Well, I'm pretty sure you guys have all heard that the microbiome is important. We know that it has an effect on a lot of different traits, both within humans, within livestock. And with the advent of more accessible and high-throughput sequencing, there's an increasing interest in evaluating these communities. And so I should put up this picture from Nature in 2010, just to demonstrate that this has been growing in interest. And I think that there's a real opportunity within the livestock community and our breed improvement or livestock improvement community to think about it a little bit more as well. So, like I said, it influences a lot of things. That's why we're interested. And it's also influenced by a lot of things, which makes it difficult. And that's part of the reason why I believe we haven't focused on it so much to date. So for example, it's influenced by things like the host's actual genetic potential. It's influenced by diet, it's influenced by additives or antibiotics, treatments of various sorts, and also just the environment in which an animal or an organism is raised. It can be influenced by developmental status. It's not so different from what Stephanie was describing with epigenetics or what we might be familiar with RNA-seq, where it is a point in time reflection of what is going on in that community at that exact stage. But despite all of that variability and all of those challenges, there's still a lot of opportunity. It's been the metagenome has been shown to directly affect multiple traits, and I have two examples up here from livestock, and that is the production of enteric methane, where we know that microbes within the rumen are what make methane, and what we're thinking about when we're talking about the pressure to reduce enteric methane production. It's also been shown to directly influence digestibility, residual feed intake, those sort of feed efficiency measures. And so those are just some examples of where the metagenome has a direct effect. But less directly, there's been evidence that the rumen or gut microbiome can have an influence on general immune response or even neurological traits. And I think that those are some of the phenotypes or traits that you might have heard more on the human side, where a lot of this research has been done in the past. So here's just some additional examples that I pulled out of some review articles. I'll be using a lot of references that are not my own, and they are not a comprehensive review of all of the literature in this space. They are just some examples to demonstrate the points that I would like to make. So for example, we have found that the rumen metagenome has a direct effect, as I said, on feed efficiency and RFI, as well as enteric methane emissions. It's also been found in dairy cattle and sheep to have an influence on milk fat and yield. And then within the human gut metagenome, that's been shown to affect a lot of different disease phenotypes, both those that are infectious in nature, as well as ones that are autoimmune. And I think that's pretty interesting. And so with that, we know that the metagenome has an effect on phenotypes. And so something that animal breeders have done, I believe these gentlemen are from Denmark, Aarhus University. They have tried to quantify that, because that's what we do as quantitative geneticists, is quantify variation. And they have termed this microbiability, and it represents the proportion of phenotypic variance of a trait that is explained by between animal differences in that community. And this should sound an awful lot like heritability, because that is exactly what it was inspired by, and that is basically what it's trying to reflect. But rather than what proportion of a trait variance is explained by additive genetic variation, we're talking about what proportion of trait variance is associated with the microbiome, or variation in the microbiome. And so just some additional examples from across a couple different species. Cattle have a microbiability for methane production, or CH4, of about 15%. Swine feed conversion ratio was found to be at about 20%. Back fat as high as 40% in swine, and then lamb live weight at about 33%. So when we think about the comparison of the proportion of variance explained by the additive genomic contribution versus this microbiome, these are fairly high. These are explaining a fairly significant proportion of some of these phenotypes. And certainly for me, again, as somebody who's interested in breeding and improvement, this is where my interest was sparked. But more than just knowing that it has an effect on trait expression, there is also some component to the metagenome that is host controlled, that is independent of both diet and environment. And that means that there is potential for us to look at the heritability of this. So I threw up this one example where they found that a core subset of the rumen microbiome was heritable in nature, as in they were able to estimate an additive control, additive variance for the control by the host of the level of abundance of specific microbes. And I didn't know that was possible before this. So in this specific study that was based out of the UK, SRUC, Mike Coffey's home institution, it's entitled Bovine Host Genome Acts on the Rumen Microbiome Function Linked to Methane Emissions. So one of the things that they found was that 17% of all of the microbial genre, or the taxonomical classification of genre, had a significant effect from the host. So that means that their abundance relative from one animal to the next could be associated with, again, that additive genomic variation. And so they were able to estimate heritabilities that ranged from 0.13 to 0.6. And again, as an animal breeder, when I start seeing heritabilities as high as 0.6, I'm starting to think about selection. That's just where my brain goes. Of those 17%, they found 29 that had a direct host genomic correlation with the host production of methane. So what that basically says is that there is host genetic control over the microbial composition that then results in differences in methane production. So we're talking about a couple different steps here to get to that final output of, or that final phenotype of methane production, but there's a lot of host control over what is happening there. And so this figure just demonstrates, I know it's quite small to see the scale, but this circle plot shows the range of heritability estimates for each of those specific genre of microbes, and it ranges from about 0.1 to 0.6. And I just throw this up to demonstrate that there is a lot of potential species in here that we could be targeting for host genetic control. In addition, they went one step further, and what they did was they looked at just the 30 most informative microbial genes and compared that to, in a simulated selection study to what would happen if you selected based on just those 30 microbial genes versus a direct methane phenotype from respiration chambers. And using the combined effect of the two of them, they demonstrated, at least within the simulation, that using even just the microbiome would increase the mitigation potential of selection. And so again, it's just to demonstrate one example of how the host genome can have an effect on the function of the microbiome, and we might care about it in regards to how that affects phenotype. So based upon all the background information I've just shared, I have generated a couple hypotheses. And again, I don't think these are particularly unique to me. My perspective on this has been influenced by a lot of conversations with a lot of different people who also have interest in this, but I'll share them with you regardless. So as the microbiome contributes a significant proportion of the variance to many traits, we may be able to use metagenomics to capture some of that additional variation that presumably is going towards the residual in our in our prediction equations right now. And additionally to that, due to the host microbiome interactions, I believe that there's a chance that we may be able to positively select for more desirable microbiomes or metagenomes. And so that leads me to where I think there's an opportunity, and that is metagenomic prediction. Again, I'm a quantitative geneticist. I really like to do predictions, and that's exactly where my mind went. And this is, again, an emerging topic within our field, and one that I'm very excited about. So there's kind of two ways to think about metagenomic prediction. One is that you can use the metagenome to predict the phenotype of the animal that you sampled from. So for example, you can use the composition of the metagenome to predict how much methane that animal may produce. Alternatively, and this is the place where I see a lot of potential, it can be thought of as a proxy trait. So for example, we know that the metagenome has an effect on methane production. Perhaps that could be used as a proxy for actual methane phenotypes to help us inform prediction of breeding values and facilitate genetic selection. And most likely, these two things would be utilized in tandem with genotypic information or pedigree information within those models. And so, as I keep mentioning, this is very new. This is the first paper published in 2013 to utilize BLUP prediction models with metagenomic shotgun sequence. And so this idea has only existed for about 10 years. This was done in Australia, predominantly using small data sets of Holstein cattle. And I think the timing of this really demonstrates where their line of thinking came from. So in 2013, this was a couple of years after genomic prediction became publicly available or commercially available within the Australian dairy industry. And so it won't be a surprise that this was very inspired by genomic BLUP, where you fit a GRM or a genomic relationship matrix. It was also inspired by rapidly decreasing sequencing costs that was occurring at about that time, particularly with short read sequencing. And then also in Australia at that time, there was starting to be a push towards evaluating methane phenotypes. And for those of us in the room who have ever attempted or thought about methane phenotyping, It's hard. And so they were looking for avenues in which they could explore proxy traits. This little study only used a maximum of 47 cattle for both reference and validation subsets, but they were able to achieve a prediction accuracy of a 0.466. And I include the little bar chart across the bottom just to demonstrate that this is a slowly building field of interest, and there's really not a lot out there yet, but it's growing. So one of the examples I want to give is this idea of holobiont prediction. I really hope I've said that right, because I've not actually heard anybody else say it to me, so bear with me. But what this group did is they evaluated residual feed intake and feed efficiency measures on about 450 Holstein dairy heifers. And so in this first model, model G, this stands for genomic or genotype, where they looked at just the host effects as we typically do in a genomic prediction model, very routine. In the model GM, they started to take into account the effect of the metagenome and the combination of those two factors. And then finally, in model GMO, they looked at not only the effect of the host DNA, but also the microbial community, and then the interaction between the host genotypes and that microbial community. So a stair-step approach, I guess you could say. And so what they found in this first chart, you'll notice this is just a regular old heritability like we would know and love. And this is at about 0.3 for RFI, residual feed intake, and at about 0.4 for, which one is that? DMI, dry matter intake. And in this next model, they presented a direct heritability, a microbiability, like I described before, and then the combined effect of the two of them. And so one kind of interesting thing I think to know is that the estimate for the direct heritability dropped, and that happened in both of these cases. It's about a 10 to 15% drop according to the article. But when you combine the effects of the two, the proportion of variance explained within that model increased fairly significantly. And then looking at the last model, which I'll put up the equation for those of you guys in the audience who like that. They looked at the interaction effect between the two. And again, using the combining the effects of this, the proportion of added or variance, trait variance explained increased quite a bit. And so I have this notice off to the side just to demonstrate that that's a fairly sizable increase of variance that's now explained. They then tested, can we use this for prediction? Does this actually do anything within our predictive models, despite the fact that we've seen that it can account for variance? And they found out that it does. You can improve prediction by including the microbial community or the composition of those microbiomes. It improved prediction accuracy. And as I pointed out before, the difference between the heritability of the straight genomic model versus the direct heritability, the authors suggested that this was because there was likely a microbiome effect that was mediated by the host genome. And that if that was not the case, there would be a difference of 0 between those two. And so this has been done in multiple different species for multiple different traits. And in my literature review, I found that in basically every instance that the metagenome was included within prediction models, that it improved the accuracy of that prediction. Sometimes it was fairly dramatic. I think for the first example, done in North Carolina, they were able to double the prediction accuracy or increase it by 100%. In the case of the rabbits, they were able to classify animals into highly productive females that had 15 litters versus those that would only have 2 with a 94% accuracy using just 53 indications of microbial variation. And then finally in sheep, they were able to explain over more than 70% of the variance of the traits of methane emissions, excuse me, and residual feed intake. And more than just those effects or the traits that we know have a direct effect of the metagenome, they also found increased prediction accuracy for weight and fleece weight, or for growth and fleece weight, excuse me. So I hope I've kind of given you an idea that maybe metagenomics can be included into our models, and maybe there actually is an effect happening that we as quantitative geneticists might be able to account for. And so what I'll do now is just briefly describe the steps that are taken to perform metagenomic predictions, again, in the event that one of you in the audience might be interested in doing this someday yourselves. So first is sample and phenotype collection. As I referenced earlier, the metagenome and the composition of it can be influenced by a lot of different external factors, and these would have to be taken into consideration with both phenotype collection and sample collection. The sample has to somehow be related to the phenotype of interest, and that said, you could pretty much sample the microbiome with whatever you want, as long as it was related to the phenotype, and as long as you were taking into consideration factors like timing. In this picture, they are swabbing the oral cavity of this cow to represent or to try to predict methane output. Next, you have to sequence the metagenome, and there's a couple different ways to do it, but these are three of the most common examples. So the first one is called amplicon sequencing, most commonly referred to as 16S. And this is a targeted sequencing approach, which means that it specifically uses primers for the 16S gene and looks at variable regions within that one gene. And then you can use those variable regions to classify the microbiome based upon genetic similarity. So the pro of this, and the reason why it's been very widely utilized, is it's very cost effective. However, the downside to this is that it's lower resolution. And both the pro and the con is the result of it being because it's a targeted sequencing approach. So it's both an advantage in that in making it a targeted approach where you're only trying to sequence a small number of variable regions, it decreases cost. It also decreases the amount of information available, and it makes it more difficult to characterize. So kind of the next step up from that is then short-read shotgun sequencing. And this is what would be done using Illumina short-read machines. Very similar to the way we would think about genomic sequencing as well. And so this is a much less biased approach than the 16S, in large part because it's not targeted. Anything that is present, or any species that are present in the microbiome, could be captured using this sequencing approach. However, one of the downsides to it is that single reads don't often span the entire microbial gene. And so again, that can create some difficulties with classification. Excuse me. And so then that leads us to long read sequencing, which would be conducted using either PacBio sequencing or nanopore long read sequencing. And so the upside to this is that because those reads are longer, they span multiple genes, and so they're much easier to classify, and they would have the highest alignment rate. However, the downside is that they're more expensive. And so what I hope to just impart upon you here is that there are multiple factors to consider, and not necessarily one right or wrong way, at least not that we've identified at this time. And that in each case, each of these have been utilized successfully for metagenomic sequencing. So as just a quick little example, I want to show you a figure that would reference how deep of sequencing you would need to perform. And so what this plot is called is a refraction curve. And so what it demonstrates is the number of species that are being classified relative to the sequencing depth. And so what you want to look for, if you were to go out to the literature to try to replicate this yourself, is the point on that curve where it becomes asymptotic, where the information being generated is where less and less new information is being generated. And so at that, based upon this, I would deduce that using long read sequencing, such as done with this figure here, that 1.5 million reads per sample was probably sufficient. So the next step is classification. And there's really two basic types. taxonomical, or functional. So in taxonomical classification, you're really just asking what species are present and at what abundance. And so there's a couple different ways to do this, but basically you classify these species down to the lowest common denominator, whatever that might be. In one of my previous examples, it was genus or genre. Hopefully you can get down to the species level, but regardless, you classify them based upon, again, the lowest common ancestor, and then classify them by abundance. And what the goal is with these classification approaches is ultimately to create a matrix of counts that shows on a per animal, per classified basis, what is the relative abundance, so that you can start to quantify differences between individuals. So at the end of a taxonomical classification, you might have a couple different metrics. It could be the actual genus or species, the taxa itself, or you might end up with something like OTUs, which is a very commonly used approach, and this stands for operational taxonomical units, or taxonomic units, excuse me. And this is basically classification based upon sequence similarities. And then another common approach is using amplicon sequence variance, which is looking at single nucleotide variation and would be very comparable to what we think genomically of as a SNP. The other approach for classification is functional, and this is asking, what do these species in the metagenome do? And can we classify them based upon that? And so in these cases, you're asking, what is the gene content that is there? And it can be further classified into functional elements, but that generally requires a lot of additional information. So in both of these, in a functional classification approach, you would need to align it to some sort of reference. And there's some struggles with having reflective reference populations or reference data sets for a metagenome. And then additional to that, you'd have to have functional annotation information available, which to the best of my knowledge is fairly limited within the livestock space. But this is one approach. And again, in each of these cases, you're making classification and then looking for abundance of how many sequence reads basically are you finding within each of those classifications on a per animal basis. And so at the end of that, you get a counts matrix. This is just an example. This only includes 12 taxa and 17 animals, but hopefully it demonstrates to you that this could be scaled up quite significantly. Excuse me. And in each of these cases, there has to be additional steps to do scaling and normalization to be able to make the comparisons between these individuals. Because what you might notice is that between each of the classifications, each of the taxa, there's potentially large variations in abundance, and these would need to be accounted for before you could use it in a way, in a matrix, as a matrix in the way that we as quantitative geneticists are typically most comfortable. And so then we get into the quantitative analysis. And how do you take this matrix of counts and do something with it? And to date, the most common approach is by creating what a lot of people are referring to as a metagenomic relationship or relational matrix. There is not one set definition or one set name for this at this stage. So this is kind of my naming convention. But it's a pretty simple calculation where X is just the matrix very similar to what I showed before, but with a natural log transformed abundances. Alternatively to fitting something like a relationship matrix that operates very much in the way that we're familiar thinking about a genomic relationship matrix would be to directly fit the relative abundances. And so in that case, like the paper example here, they fit each of the abundances of the various microbes as individual univariate models. And so this approach would be very akin to something like SNP-LUP. And then you get into predictive modeling, and I won't go too far into the weeds with this, but I want to highlight a couple key points to consider when you think about the predictive modeling. One is, what are you trying to predict? Is it a phenotype? Or are you using the metagenome as a proxy trait for a breeding value? And in practice, this doesn't look so different, but it does change our interpretation. There is also no standard at this stage about what is the best approach for modeling. And so linear, Bayesian, machine learning, all have been tested and all of them have had some success, but there is not yet a accepted status quo, so to speak. And in where I see the most potential is fitting both a GRM and, as I referred to, a metagenomic relationship matrix simultaneously. However, there are still some limitations with modeling this approach, where you can model both of them simultaneously, but it does produce separate variance estimates. And so you can estimate then microbiability and heritability, but to date, I have not seen anything where you can estimate the total effect all at once, or the combined effect into one single estimate, excuse me. And so, maybe I've convinced you that there's some value to this, that there's some interest, but I'm also saying, hey guys, let's do more sequencing. And so really, where is the value to metagenomics? And where I think it is in capturing additional variants for low to moderate heritability traits. Those are the traits that already have been shown to increase in prediction accuracy the most with the inclusion of metagenomics into the model. And I think this is especially true for difficult to measure traits. So just as a rough example, if we were to use metagenomics as a proxy for one of these more lowly heritable, difficult to measure traits, what would that kind of look like? And so I roughly estimate that the cost of a long read metagenomic sample is about $100. A feed efficiency trial, though, is about $750 a head. 1 year of missed cow production due to infertility is estimated to cost about $900. And I don't have a per animal estimate for disease, but for example, BRD, the cost of BRD prevention, management, treatment, and herd loss costs the industry about $1 billion per year. And so these are some lowly heritable traits that I think we can see how the metagenome could be playing a role, where maybe there is opportunity to utilize metagenomics to improve prediction or to serve as a proxy trait. And with that, saying that this could be a proxy trait, I am absolutely not trying to say that we need to stop phenotyping. I'm pretty sure my coffee would come around and get me if I did. But it is maybe an avenue to increase the size of our reference populations. So one example of this, working with Elizabeth Ross at Queensland, she performed a simulation study to sort of serve as a case. And so in this instance, she was interested in evaluating what would it take if we wanted to facilitate genetic selection to reduce methane production in Northern Australia. So some of you may be aware that in Australia, there's extremely strong social and regulatory pressures on the beef industry to make an effort towards reducing enteric methane. And one way that they're exploring to do that is through genetic selection. And with it, methane being a fairly lowly heritable trait, this will take a large reference population, and that's expensive. So she made some assumptions within the simulation study that I would like to use as an example, and assumed both a large effective population size, in part because in Northern Australia, they're very large admixed crossbred herds with unknown breed compositions. So it's pretty safe to assume there's a lot of variation, genomic variation. She also assumed a heritability of methane of about 17%, and that the genomic correlation or genetic correlation between individual animal methane output and then the predicted methane output estimated using metagenomics was about 0.66. In her lab, the cost of metagenomic sequence sample is about 160 Australian dollars. And recently it cost her over $2,700 to get a single animal evaluated using a green feed machine. For this simulation, she wanted to know how many animals, or what would the cost be to get a reasonable prediction accuracy to be able to facilitate this within her home country of Australia. And so she utilized the equation that I have put up here, which she scaled by that genetic correlation between the direct and the metagenomic predicted phenotype for methane. And then the scenario that I want to describe to you today is one where all animals have a metagenomic profile and 5% of them have a green feed phenotype. And I want to preface before I show you the results of this, this is just an example, it's a case study, it's not super sophisticated, and I only mean to show it, not as something that necessarily will occur in this country, but just to show the potential value. So again, in a scenario where all animals have a metagenomic profile, but 5% have a green feed phenotype, what I think we should look at, especially over in a scenario where there's a whole lot of genomic variability, is at a cost of approximately 5 million Australian dollars. So say the Australian government gave them $5 million instead, maximize your opportunity, maximize the size of this reference population. The predicted accuracy that would be achieved with that $5 million is significantly higher when using microbiome information in combination or in this case by itself. And these practices did not take into account the interaction between these two, which is why the microbiome only estimates are a little bit higher than the combination. We would expect them to go even more, but the point still is that say at the $5 million mark, the cost to, or the accuracy that could be achieved is much higher using metagenomics. And this is true across all the scenarios here, which were based off of effective population size and roughly correlate to these breeds within the Australian beef herd. So I've described some examples of where metagenomics was utilized to inform prediction for residual feed intake or for other feed efficiency measures, as well as reducing enteric methane production. But where I see some additional perspective or additional opportunities, as I somewhat hinted at before, is in the improvement of disease resistance. I could easily see how we could try to predict improved resistance to BRD using the nasal microbiome, or pink eye resistance using either the nasal or ocular microbiome, or maybe even venereal disease, sampling either vaginal or seminal microbiomes. I also think that there's potential to utilize this information to improve reproduction. I could see ways in which we could test and evaluate whether or not this would improve our prediction accuracy for conception rate or reduce pregnancy loss or embryonic loss. And I'm aware of some early studies coming out of Australia investigating the effect of the microbiome on IVF success rate and being able to predict what will happen. So despite the fact that I think there's a lot of potential, there's still a lot of challenges. This is early in development. That's why this is a fairly general overview. And some of the difficulties that still exist is that it still requires a fairly large reference population size. This is still a quantitative trait. If we were to think back to those heritabilities that I described at the beginning, a lot of them were still in that low to moderate level. And so that means we still would need fairly large groups of animals with metagenomic profiles in order for them to be useful. And currently, because this is still very much in the research and development phase, there's a pretty pretty big P versus N problem, which basically just means that there are in a lot of the data sets, there are more classifications, or P, versus the number of animals represented, which is N. And this should sound fairly similar to those in the room of some of the struggles that were incurred at the early stages of using genotypes, where we had more individual SNP than we did animals. There's also some real questions about data interpretation. Again, I'm not a microbiologist, so this is probably a space in which I also struggle. But the effect direction of metagenomic predictions is not always known. So for example, is the microbiome affecting the trait, or is the trait the phenotype affecting the microbiome? So with enteric methane production, we know that the rumen microbiome is what is producing enteric methane. But there's examples of autism in humans where the metagenomic profile has been associated with autism spectrum disorder. However, they have found that it's not that the metagenome or the gut microbiome is having an effect on disease expression. It's that the expression of the disease changes the dietary preferences of those individuals and affecting the microbiome. However, if we were to assume the other direction, especially with some of these more sensitive phenotypes, we could really be doing more harm than good. And so I think it takes a little extra element of consideration, especially when we're used to thinking about more linear additive genetics. There's also a lot of struggles with the timing, sampling, and the reporting of the metadata associated with this, so that you can accurately compare results between studies. because there are so many different external factors that can influence the microbiome, and the way that we classify the microbiome or the metagenome can also have a role on our ability to compare between studies. There's probably a need to increase the amount of metadata that's reported. And so up on the screen, I have shared a very long list of the various places where I think there are ongoing research questions. And this is where I really see a lot of opportunity. We don't have a great understanding of the underlying biology and how is the host genome actually inducing an effect on the microbiome. I'm not sure. Maybe it's passage rate of the rumen affects the microbial composition, and it's actually the passage rate that is heritable. I don't know. So there's questions about that. There's a lot of work to be done, I think, on optimizing the modeling approaches and the estimation of the relative differences in metagenomic profiles between one animal versus the next. At this stage, I think it's pretty naive and rudimentary, and there's a lot of room to grow. And with that, there's a lot of room to grow on the bioinformatics side. What are the actual requirements for sequencing? How should we classify that metagenomic data? Do we have sufficient reference data sets in order to do that effectively? What are the interactions between the host and the metagenome? And we don't actually know if we can make these selection choices and influence the metagenome. So far, for the rumen metagenome, for example, we as scientists, producers, et cetera, have not been very good at manipulating it in a consistent way that incurs long-term stabilized effects. Can we actually make selection choices that will influence this? I'm not sure. It seems like it would. That's my hypothesis, but it should be tested. So in summary, I hope I've convinced you that the microbiome has potentially very large effects on certain phenotypes, specifically those that are directly related, as well as others that are low heritability, and that between animal variation in the metagenome can be utilized to increase our prediction accuracy, or at least the proportion of variance explained within our predictive models. And so I think that this leads towards opportunities to use metagenomic prediction for selection, or to use metagenomics as a proxy trait. And the thing that makes me most excited is the opportunity. There's lots of room for improvement, and for somebody like me who's a scientist, that looks like an opportunity. And I hope it does to some of you in the room as well. And so with that, I need to acknowledge the many people who have helped instruct me, inform me on the topics that I've discussed here today, in particular, my collaborator, Elizabeth Ross from the University of Queensland, as well as her postdoc, Chen Yong, and then my colleagues at USMARC, Larry Keane, Jim Wells, Tara McDaniel, Tim Smith, Kristen Kuhn, and Sun Shin. They have had multiple discussions with me that have helped inform my perspective on this topic. And then also from the University of Nebraska, Lucas Fernando. So members of this list have previously generated multiple metagenomic data sets associated with different projects and different topics. And we hope to use some of that historical data to test some of the theories that I've described here. But I also hope that if you guys have access to those data sets, that you might do so as well. And so with that, I would be happy to field any questions that you guys may have.